Hi everyone, my name is Chelsea Gerkink. Today I'm going to talk to you about neutrophil-specific antigens, and I've written everything down, so I'm just going to show you the pages that I've written. Thanks. So the title of my assignment is called Neutrophil-Specific Antigens, Immunology and Implications in Transfusion Medicine and Blood Disorders. So before I talk about the neutrophil-specific antigens, I kind of just wrote down some general questions that I thought would be good to go over before talking specifically about that. The first one is what is a leukocyte? And as we all know, a leukocyte is a cell of the immune system that is involved in protecting the body against infections. So any foreign invader in our body, our major cells in combating those foreign bodies are cells like lymphocytes, granulocytes, monocytes, macrophages, and of course the granulocytes include eosinophils, basophils, and neutrophils. And so my second question is, how do these leukocytes impact transfusion medicine? So, leukocytes obviously contain antigens on their surface that can elicit an immune response when patients receive donor transfusions. And actually, so this is going to the article now. There was a guy named Tom Brittingham. I can show you a picture right here. There he is. He actually was a crafty guy, and he decided to inject leukocytes into his bloodstream from a patient with chronic myelogenous leukemia. And he discovered that when he controlled the number of lymphocytes transfused and the amount, and over time, he noticed that he was getting severe reactions, including high fever, vomiting, hypotension, bilateral lung infiltrations, aka trolley. And basically, these reactions represented the production of leukoagglutinins, aka antibodies, that he was producing against these foreign leukocytes that he injected into his bloodstream. So I just did a little drawing here. This is Tom Brittingham's leukoagglutinin that he's created against these donor lymphocytes. Identification of neutrophil-specific antigens, the focus of this article. So there were four, it starts talking about how there were four infants that this team followed. The first infant died of sepsis, and then the three other infants presented with severe neutropenia at birth. And when they actually looked at the slides of these infants, they found no neutrophils on the slides. They soon discovered that these neutrophils were actually being destroyed by the mom's neutrophil antibodies that she had created against the baby. They discovered that the antibody in question could only be absorbed by neutrophils and not by other blood or tissue cells. So here's an example. The mom created this anti-neutrophil antibody, and it started to attack the baby's neutrophils and destroyed them. So they decided to diagnose and term this presentation as alloimmune neonatal neutropenia, also known as AN. The length and resolve of AN is that it lasts as long as the maternal antibodies remain in infant's blood. So the neutrophils will continue to be destroyed until the maternal antibodies are removed in circulation by the baby. And then the presentation is skin infections, omphalitis, and the main presentation is that AN occurs in the first pregnancy. This differentiates it from erythroblastus fetalis that just usually occurs in multiparous women. This will also, AN will actually present in first pregnancies. So the second part of the article really goes into the NA-NB system. So I'm going to talk about that. Just for key terminology, N equals neutrophil, A equals the first presumed locus for that antigen, so B will be the second presumed locus for the antigen, and then the number is the allele that it's found. So in NA, the neutrophil antigen, you have NA1 through 4, and then you have NA null. You can see the frequency of the NA and the NB. The NB is pretty high, 97% have the NB antigen, whereas only about 56% have the NA. So if you remember, I was talking about the babies before. NA1 was discovered in those babies. So let's talk about NA. NA is the receptor for IgG1 and IgG3. NA antigens bind only to immune complexes, not to free immunoglobulins. Meaning that if a red cell and an antibody complex together, the neutrophil antigen would bind to those, and then they would be destroyed. So you have to have a reaction of something else to bind to get that neutrophil to be destroyed. And then the NA null patients, only about 20% of them developed frequent infections in a study that was done. So not having the NA doesn't necessarily mean you're going to be immunocompromised. And then NB is an adhesion molecule. It's also known as CD177. There wasn't too much information about it in the rest of the article. It mostly talked about the molecular framework of the NB antigen, which was pretty interesting. It discussed TRALI and its association with neutrophils. So TRALI stands for transfusion-related acute lung injury, and it's the most common cause of transfusion-related death. So what happens in TRALI is that it's typically implicated in platelets or plasma, mostly plasma, and the donor plasma is transfused into the patient, and in the pulmonary endothelium, you have your pulmonary endothelium right here. You have some unstimulated neutrophils, and so what actually happens is the donor's antibodies, they actually sensitize to these unstimulated neutrophils. They activate them, and the neutrophils undergo something called oxidative burst, which releases toxic substances that damage the pulmonary endothelium. As soon as the cell is damaged, this results in capillary leak and pulmonary edema, so you get your classic bilateral lung infiltrates. And the implicated antibodies in TRALI are human leukocyte antibodies and human neutrophil antibodies. Now I'm going to talk about the role of neutrophil antigens in TRALI. As I've learned in the lab and also in school, TRALI is very complex, and it is actually pretty unknown how it occurs in the body. So this article not only talks about how TRALI occurs in the body, but a possible hypothesis about how the neutrophils play a huge role in TRALI. So the main line here is that TRALI may be considered, the antibodies may be considered chemotactic factors and neutrophils as the chemotactic receptors. So there's some type of chemotaxis going on, and some of the major players in TRALI would be complement here, and then someone also discovered that von Willebrand's factor was involved, and finally PCAM, as we've all known. This is kind of like that sticky molecule. So, of course, if this hypothesis would have to be confirmed with animal models, but it's very interesting that they thought about how this is occurring and not just how to diagnose it. Okay, I'm going to talk about the methodology for leukocyte antibody detection. In other words, the testing in the lab that we do to discover neutrophil antigens of the patient. Basically, overall in this article, you can kind of tell that neutrophil antigens are up and coming. We don't know a lot about it, and they're not very specific and sensitive yet. So basically, neutrophil agglutination testing requires live cells. So this can kind of deter and kind of make testing difficult. And some different tests include immunofluorescence, monoclonal antibody mobilization of granulocyte antigens, ELISAs, and genotyping. And one interesting part about the genotyping is that there have been some patients that have produced an NB1 antibody, but they actually carry the NB gene. So there's some discrepancies there, and they've really talked about how it has to do with the protein synthesis and how it's not perfect in testing yet. One also talks about a serious issue raised that many clinical investigators and clinicians have expressed that neutrophil antibody testing has little or no value in the diagnosis or management of neutropenia. And this can be difficult for the patient because I'm sure they'd want to know what's going on, but also perhaps because the testing isn't necessarily up to par. It's going to be expensive and the clinicians aren't really going to know what's going on. So the article kind of discusses a solution for this problem right here, is that experienced neutrophil immunologists have developed more advanced technology, and so they recommend kind of going to reference labs for this information and not trying to depend on the clinicians. So this is the conclusion of my article. Basically, it talks about what I've been talking about throughout the presentation that I did today, that neutrophil immunobiology actually needs more investigation. This not only includes understanding the pathogenesis of TRALI, but also in cases such as AN and AINI. Also, we want to standardize more of our testing so that clinicians can have a direct test they can go to diagnose Anne and AINI and trolley, because a lot of it is really unknown. So that's the best part of science, is discovering the unknown. So thank you so much, and I hope you've learned a lot about neutrophils today.